Meiosis. Meiosis starts with interphase. During this time, there are centrioles and a nuclear membrane. The X-shaped chromosomes inside the nuclear membrane are color-coded and homologous to each other. Each chromosome has a pair of sister chromatids. The centromere is right in the middle of the chromosomes. During prophase 1, spindle fibers first start to appear. The nuclear membrane starts to dissipate around the chromosomes while the spindle fibers still grow. During late prophase 1, the chromosomes start to move closer to the center. Also during prophase 1, synapses and crossing over happens. This switches information between homologous pairs as shown. The next step of meiosis is metaphase 1. The spindle fibers grow toward the center of the cell, eventually connecting to the centromeres of the chromosomes. In anaphase 1, after the spindle fibers connect to the centromeres, the homologous pairs are separated on ends of the cell towards the centrioles. During telophase 1, the duplicated chromosomes are now on opposite ends of the cell. A nuclear membrane is now forming around the two groups of chromosomes. Telophase 1 differs from telophase in mitosis because there are still duplicated chromosomes, and the daughter cells are not identical. Later during telophase, cytokinesis starts. The cell membrane indents and eventually cuts off to make two diploid cells. There's no interphase between cytokinesis 1 and prophase 2, so each new cell will have half the number of chromosomes as the original parent. Only difference between metaphase 1 and 2 is that metaphase 2 has one line of chromosomes. Anaphase 2 is like anaphase 1, but now the duplicated chromosomes are separated from each other and moving toward the centrioles at opposite ends of the cell. Telophase 2 is exactly like telophase 1, but telophase 2 has half as much chromosomes and genetic information. Meiosis is sometimes called reduction division because of this. The genetic material is reduced. This is also what differentiates meiosis products with mitosis products. These cells can make another living organism. Again, later in telophase, cytokinesis starts, indenting the ends of the cell and eventually cutting off the two nuclei. This is also the cell division that makes the cells haploid. If every gene was homozygous, then there would be no genetic diversity between the haploid cells. The final product is now haploid cells.